All right, class. So we have already looked into the processes of glomerular filtration that take place at the glomerulus and Bowman's capsule. And we have also talked about the process of selective reabsorption that take place in the proximal convoluted tubule. Now we will focus on the mechanisms that work on the descending limb of the nephron loop and the ascending limb of the nephron loop. Sorry. Okay, to start with, I would like to I'm sorry, class. To start with, I would like to highlight few things. First, remember that the osmolarity of the blood in the glomerulus is about 300 milliosmos. Same is the osmolarity of the filtrate here. The filtrate is 300 milliosmos. At the end of the proximal convoluted here, the concentration of the filtrate is 300 milliosmos as well, and that is equal to the concentration of the fluid in the interstitial space. So in the interstitial space also, the concentration is 300 milliosmos. Okay, that's something to know. And we have already seen how salt and glucose and water, most of the water, get reabsorbed back from the proximal convoluted tubule. So we know that salt, glucose, a lot of water, vitamins, all these things get reabsorbed back into the system from the proximal convoluted tubule. We know that. Also remember, about 70% of the water is actually gets reabsorbed from the PCT. All right. Now here you do see the nephron, but on top of that you see these weird light blue colored lines. You are like, Rupa, where did they come from? Well, I want you to know one thing. Along the loop of Henle, in the medulla of the kidney, if you remember the medulla part of the kidney, there exists a concentration gradient. So I will just draw the concentration gradient here. So this part is 300 milliosmos. This is 400. This is 600. This is 800. This is 1000. And this is 1200 milliosmos. I'm not writing milliosmos all the way down. So what do we see? We are looking into the interstitial space in the medulla, right? So this is the interstitial space in the medulla where I have written these numbers. And we see that as we go down deeper into the medulla, the solute concentration gets higher and higher and higher and higher until it reaches 1200 milliosmos. Right? And we have also known another thing, if you remember from my lecture on day 1 and day 2 of class, when we talked about the tonicity. Remember, we talked about the tonicity. I mentioned one thing: solutes suck. Solutes suck. Really important. So as we go down the medulla, as we go this way, do you see that there is increased solute concentration? Very good. Now let's look at the tubular fluid now. I'll mark it with a different color. So the tubular fluid is here with 300 milliosmos. Now the fluid comes down, 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 down the descending limb. As the fluid comes down the descending limb, where do you have, sorry, before that, remember, the descending limb of the loop of Henle is permeable to water and water only. I will write this down. I'll make a note here. Descending limb of the loop of Henle is permeable to water only. It cannot transport any salt or any other solute because it doesn't have those transporters and those pumps. No, nothing. These are just super simple cells. So since these cells are, so the cells are lining here, right? Since these cells are permeable to water only, obviously, based on the concentration gradient in the medulla, water will leave. So water leaves. Class, I want you to think about this. So the fluid comes down, right? The fluid comes down and water is leaving. We are just, the solutes that are in the interstitial space, the solids are sucking out water. So water is leaving the descending limb of loop of Henle. As water leaves, what will happen to the concentration of the filtrate? The concentration of the filtrate gets higher and higher. The filtrate gets super concentrated until, we started with 300 here, right? So it becomes, the concentration becomes 400, 600, 800, 1000, and 1200. So you see how the fluid is becoming, how the filtrate is becoming more and more concentrated. Very good. So here it's about 1200 milliosmos. Okay. So what will happen next? Before we see what happens at the ascending limb of the loop of Henle, I want you to remember that the cells lining the ascending limb, these cells, they are waterproof cells. Can I write it? These are waterproof cells, meaning they will never ever allow water to either enter or leave the cell. Therefore, so this fluid, the filtrate, now moves to the ascending limb, and as it passes through the ascending limb, remember it's water impermeable, it is waterproof. So at this point it only pumps out salt. It pumps out actively by active transport. It pumps out sodium chloride, potassium. All these salts are actively pumped out. Now I want you to think about this. First, in the descending limb, you lost water from the filtrate. As you lose water from the filtrate, obviously the filtrate gets more concentrated. Now as the filtrate moves up the ascending limb, it is impermeable to water, but these cells are actively pumping out salt into the interstitial space. Therefore, what is happening to the concentration of the filtrate? You are now taking out salt. So are you diluting it or are you concentrating it? You are concentrating it. Sorry, you are taking out salt. So the filtrate is now getting dilute. So here, it will go from 1000 to 800, 600, 400, and ultimately when it reaches the distal convoluted tubule, the filtrate will have a concentration of 100 milliosmos per 100 milliosmos. Okay, that will be the osmolarity of the filtrate when it reaches the distal convoluted tubule. Now, I did mention that water is being pulled out of the descending limb, and then salts leave the ascending limb of the loop of Henle. But after they leave, where do they go? Do they just hang out in the interstitial space? No. Remember vasa recta and peritubular capillaries that wrap around the nephron tubule. So these water and salt will be transported from the interstitial space back into the capillary that surrounds these tubules. All right, so I'll stop here and then start with a new video.